So our next speaker, which I'm very excited to introduce, is David Berry, and he's assistant professor at the University of Vienna at the Department of Microbiology and Ecosystem Science. And the project that David is going to talk about today is a project or an effort that JGI is very excited about. And we would, specifically as we move in our strategic planning from sequence to function. And this involves a methodology that I would put into the category of function-driven genomics, which is a newer box that we have. But essentially, it helps us, or it enables projects to not just sequence more of anything, but really go into environments, specifically more complex environments, with a targeted approach to pull out organisms based on a functional or phenotypic trait that we really care about and that we're interested in. So in that regards, David has a collaboration with Michi Wagner at the University of Vienna to explore single-cell Raman microspectroscopy to specifically isolate labeled cells. So thank you.
Good afternoon, everyone. My talk is a little bit different, I think, from a lot of the other things we've heard at this conference. But I just checked through the slides, and I do have some sequencing data, so you should feel at least comfortable for part of it. So what I would like to do in the next half hour, basically, is to try to convince you that single-cell isotope probing via Raman microspectroscopy or nanosims is a very powerful tool for looking at the function, the activity of microbes in the environment. So we've heard a lot about microbial dark matter. I just wanted to point out that actually we've been illuminating dark matter since the mid-90s with fluorescence in situ hybridization. This is a fish micrograph of a wastewater sample, and all of these are different groups of uncultured microbes. And the big question then, and still to a large part is today, what are these microbes doing? And the meta-omics revolution has been really an exciting progress and produced a lot of very interesting results to answer this question. And we've heard a lot about this at the conference. But even though this is very important and it's a necessary development, it's not sufficient by itself to really look at activity in the environment. Why is that? Why do we need other tools? Well, metagenomic data can generate hypotheses, but these can be wrong or incomplete based on the quality of the annotation, based on proteins with multiple functions, like moonlighting enzymes, for example. Also, the existence of a transcript or a protein is an indication, but it's not really a direct proof that an enzyme is active in situ. And we heard in Martin Ackermann's talk yesterday about functional heterogeneity within even a clonal population. And if you think about microdiversity in the environment, and also of the complex microhabitats in environments, it's clear that this could be a very important process, and we can really only study it in situ on the single-cell level. So this is the outline of my talk. Basically, I'm going to give you an intro to single-cell isotope probing and introduce two methods. We've heard a little bit about nanosims, but I'll try to describe it in some more detail. And even though it's a technical session, I'll give you two examples of how we're using, how these technologies can be used. So single-cell isotope probing, this is sometimes put very simply, who eats what. So we're often trying to figure out who are the key players, the key microbial players in some process. And the first step with this is labeling biomass. And you want to label biomass with an isotope label. This can be a stable isotope, which is a non-toxic isotope, or a radioisotope. The advantage of the stable isotope is obviously that you can apply it in the environment or in the body without any kind of health concerns. So for example, if you are interested in looking at xylan degraders in soils, you could add a 13C labeled xylan to your environment and then allow the microbes to do their process, degrading the xylan, taking up some of that 13C and building up biomass with it. Then you would fix the sample, and you would go in and identify target populations using fish, targeting specific groups that you're interested in, and then you can detect which organisms, which cells here that are fish labeled have taken up the isotope using one of a variety of tools, and today I'll only talk about nanosims and Raman spectroscopy. So this is a Raman microspectroscope. It's not a very big instrument, actually. This is a typical lab unit. Here you have lasers and some optics. This is a typical fluorescence microscope, and your sample goes right here. It's pretty easy to operate. This is actually a rather large unit. You can miniaturize these. You can also make them portable. You can use fiber optics, so you can take them out into deep sea. You can also, if you're interested in human microbiome, you can use the fiber optics for a colonoscope, but I don't have any images of that here. So this is really a versatile technology. And yeah, what I'd like to say, that the two key things here is, one is that it's non-destructive. So you can do live cell imaging. You can do this in media. And also, it's very fast. So if you have an optimized system, you can make a Raman measurement of a single cell in under a second. So the principle of Raman microspectroscopy, basically, it takes advantage of the so-called Raman effect, which is the inelastic scattering of photons. So without getting too in detail about the physics, basically what happens is when you shine a laser light onto a microbial cell, the photons from that laser strike the bonds in the cell. And occasionally, this causes the bonds to get excited and to have different shapes, out-of-plane deformation, stretching, ring breathing. And when the photon then is scattered, the departing photon has a different amount of energy as the incoming photon. This is called Stokes scattering. And that's because some of the energy is left with this bond. And what we're measuring with the Raman is basically the photons and the shifted energy of the photons. So the amount of energy that's left, basically, it depends on the atoms in this bond and also the type of vibration in the bond. And if you do a Raman imaging of a single cell, so here we have an E. coli, and the Raman laser is right here in this green dot, you get a very complex spectrum. Of course, a microbial cell has a lot of different types of bonds, a lot of different types of atoms, and for a typical cell, you get about 1,000 peaks. Some of these are clearly differentiated, and some of them are more stacked together. But you can see that you get compound classes that are characteristic: nucleic acids, carbohydrates, proteins, lipids. Phenylalanine, because it has an aromatic ring, has a very distinctive Raman signature, and we'll take advantage of this later. I'll show you in a few slides how we take advantage of that for stable isotope probing. Now, not only can you measure these kind of basic constituents of a cell, but you can also measure if a cell has storage compounds, because abundant compounds like storage compounds, cytochromes as well, carotenoids, these give really distinctive signatures in the cell. So if you're interested, for example, in exploring an environment for mining for novel storage compounds for bioenergy purposes, for example, this would be, Raman microscopy would be one way to look for something new here. And just as a quick example of this, in a collaborative project last year, members of the Raman team worked with a group of Marcel Kuipers on a two-member consortium, a delta proteobacteria and archaea in the oceans. And this consortium reduces sulfate and oxidizes methane. And it was always thought that the delta proteobacteria were responsible for the sulfate reduction. But when we looked at this sample in the Raman microspectroscope, you can see here in red are it doesn't come up so good here, but in red are the archaea. When we looked at the cell spectra of the archaea and the delta proteobacteria, we found that it's actually the archaea that have elemental sulfur storage in them. And it turned out that very unexpectedly, the archaea are actually responsible for this sulfate reduction. And we were able to directly show that this is the case with the Raman. So if we want to use Raman microspectroscopy for stable isotope probing, I mentioned that we have a very strong band in phenylalanine here at 1003 wave number. And what happens is that because the Raman shift that we observe is dependent on the mass of the atoms involved in the molecule, once 13C replaces 12C in the phenylalanine ring, this ring is able to take more energy from the photon, and we observe a shift from 1003 to 967. And what we can do is to take the ratio of the 967 peak to the 1003 peak, and then we have really a quantitative metric to measure how much 13C a cell has taken up. This is great if you have a compound labeled that you're interested in, but if you're working with complex compounds, complex carbon degradation, for example, or you have another question that's not related to a specific compound that you can label, you need an alternative method. And something we've been really active in is to develop activity markers that aren't dependent on labeling a single compound. And what we're really excited about, we've come up with, is using deuterated water, so D2O, in an incubation. And this takes advantage, basically, of the fact that when cells are active, they're producing new lipids. And these lipids, the lipid biosynthesis pathway uses NADH. And when the NAD is reduced to NADH, it's using hydrogen from the water. Now, if we replace some of the water with deuterated water, now we're forming NADD instead of NADH. That deuterium is then transferred onto the lipids, and we have CD bonds where we never have CD bonds naturally in nature at any real concentration. So if we grow E. coli in D2O, you can see here, this region is where the CD bonds come up. And this gives us really a nice general activity marker based on lipid biosynthesis. And we've tried this out with a lot of different types of cells, with a lot of different physiologies, autotrophs, archaea, heterotrophs, and basically in all of them, we're able to see the CD peak come up with cell activity. So we have here a really nice activity marker, and I'll show you in the second application today how we're able to use this to study nitrifier communities. So nanosims, this is the other technology that is really powerful for single-cell stable isotope probing. This is high-resolution secondary ion mass spectrometry. You've heard a little bit about it in a few of the talks, and you've probably always, with every talk, seen this beautiful image that comes from Mickey Wagner's review, a very good review on the subject. But briefly, what it is, I'll show the instrument first. This is it. It's a gigantic machine. It sits in a clean room. It needs a full-time, specially trained operator. It's incredibly low throughput. It's everything, basically, that the Raman isn't in this sense. So why work with such an awful machine? This is because this high resolution part of the SIMS is really special. So it has, it's high resolution in two senses. One, it has a very high spatial resolution, so you can do subcellular imaging down to 50 nanometer resolution. So you can even image a single flagellum with this method. And the other reason that it's high resolution is that it has a very high mass resolving power. So it's able to resolve isobaric species. So you can resolve 12C versus 13C. And it's not just carbon and nitrogen here, but you can also measure heavy metals, for example, if you're interested in how heavy metals may be compartmentalized in a cell. So basically, it works in this way, that there's a primary ion beam, a high energy beam. That's in this tower. You put your sample in a vacuum here, and you sputter your cells with the primary ion beam. This produces secondary ions, which are then routed around this mass spectrometer, and you can measure 7 different mass sizes in parallel. And now I'll give you an application of how we've used the nanosims to answer a question in gut microbiology, and this is to look at which organisms are important host compound foragers. Most bacteria in our gut are making their living breaking down things that we've eaten and essentially are feeding them. But there is a subsection or a clade in the gut that makes their living by breaking down not what we eat with our diet, but actually what we secrete into the gut to provide as a barrier. And these are the host compound foragers, or the mucus degraders. So along the gastrointestinal tract, you have your mucosal tissue. And out here in pink, you have all of the bacteria. It's an incredible ecosystem, very densely packed. And these, we're separated basically from our microbiome by a very thin 50-micron layer of slime, simply put. The mucosal tissue is basically secreting this mucus. It's a complex mixture of glycoproteins continuously, and the gut microbiota is continuously degrading it. So what we have is really a state in dynamic equilibrium. When we lose this equilibrium, such as is the case with this genetic knockout, there's inflammation occurring in the longer term cancer. So this is really an important barrier for health. But we really know very little about who it is, who are the key players in mucus degradation. And it's a difficult area to study because you have different compounds being produced. So what do you label is the first question. And then it's a very tight symbiosis. So how do you introduce a label without totally disrupting the system? So we decided to take a step back and basically let the mouse do the hard work for us. We gave a precursor amino acid, 13C15N, labeled threonine, into the blood of mice and let the epithelial tissue basically produce mucosal proteins and secrete them itself so that we could study the system without disturbing it. So when we do this experiment, we see basically that the threonine, after injection, decreases over time, as we would expect. It's being used all around the body. Also, it's appearing in the tissue, and some of it gets fixed in the tissue over the order of a day. And this is where it gets interesting when we look in the lumen, so in the center of the gut, we see after some delay, after a few hours, we see our label appearing. And not only is it appearing, but we also see the 13C appearing in the fermentation pools, acetate, propionate, and butyrate, indicating that the bacteria are actually already, they see these labeled compounds and are already breaking them down after just a few hours. Now, if we go in with the nanosims, we can actually image this label transfer. So here we have the epithelial tissue. And in here we have the goblet cells that are producing the mucus. And out here is where the bacteria are sitting. And you can see in this isotope deviation map, these hotspots indicate where our label is. So they're accumulating here in the goblet cells, ready to be secreted. And here at 8 hours, we can also see hotspots out here in the gut. And these are bacteria that are labeled. So now the next challenge is to figure out who is it that's labeled. We see that there are cells, but how do we identify them? And for this, we use fluorescence in situ hybridization, targeting species and genus level groups in the gut, so abundant groups. And essentially what we do is we take a sample, we do a fish staining, and then we mark the area we're looking at with a laser. And then we take that very same sample and put it in the nanosims. We use the laser markings to find exactly the same spot, and then we do an isotope imaging in the nitrogen channel here and in the carbon channel. And you can see, for example, this blue cell is labeled in nitrogen and also in carbon, but this blue cell sitting next to it is not labeled. And now I summarize really a lot of work. We've measured hundreds of cells with 5 major populations. And each dot here is basically one cell that we've measured with the nanosims. And in the end, so the punchline is that we identified the 2 major groups that are responsible for degrading these host-secreted proteins. These are Akkermansia muciniphila and Bacteroides acidophasians. These 2 together, given their abundance, we say are responsible for almost all of this activity. So we said, this is great. We've identified the 2 major groups. Now let's try to simplify the system a bit. We take these 2 species and add them into a notobiotic mouse. This is a mouse that has just 4 known strains. So a low complexity system that we can throw metatranscriptomics and everything else at it and learn a little bit more about what it's doing. So we put them into these low complexity mice, let them colonize. They were very happy there. And then we repeated this experiment. And we were really surprised to see this is their activity in the high complexity system, so the native system. And this is their activity in the low complexity system. And this is strikingly different. What we think is happening here, and it was totally unexpected, but what we think is happening is that these strains in the high complexity system are really only competitive foraging on this one niche, whereas in the low complexity system, they're missing a lot of competitors that they have here, and they're able to expand their niche into dietary compounds that are easier to degrade. And the point is that if we had started in this low complexity system, we never would have identified these as the major mucus degraders. And so this, really the punchline is that this is why it's so important to study microbial populations in the wild, to look at really what the activity is in the wild. Now, in my second example, I want to talk about exploring genomic and physiological diversity, microdiversity within nitrifiers. And this is where we use Raman microspectroscopy. So if you don't know about nitrification, this is a very important process in the environment and also in the engineered environment, so everything from agricultural soils to wastewater treatment. Nitrification is a two-step process. It's catalyzed by ammonia oxidizers, bacteria, and more recently we know archaea, responsible for oxidizing ammonium to nitrite, and nitrite oxidizing bacteria further oxidize the nitrite to nitrate. And you can see that this is a micrograph from a wastewater treatment plant, and you can see that the ammonia oxidizers and nitrite oxidizers tend to grow in this microcolony structure and also tend to grow together in microcolonies, which makes sense since they're metabolic mutualists. Now, we've been studying nitrifiers for a long time in our department, and one thing that we've observed quite frequently is that there's really a large amount of microdiversity within both ammonia oxidizers and nitrite oxidizers. We see this in lots of different environments, but this is one study where Chrissy here, she's a little cut off, but she was studying just different clusters within a single lineage of Nitrospira in a wastewater treatment plant over several years. And what she observed with sequencing and also with fish was that these different clusters, which are very closely related on the 16S level, were consistently co-occurring and also co-occurring stably over time. And this is a big mystery of why this would be. If there's really only one niche that all of them are competing for, how are they all able to survive? We have some indications from microautoradiography that there could be some substrate partitioning, so one cluster tends to use formate more preferentially than others, but it still is a rather open mystery. And this is what's kind of motivated this project that we have with the JGI, the single active nitrifier microcolony genomics pipeline. Essentially, what we do here is we take an environmental sample, so here from wastewater, incubate it with ammonium and bicarbonate, stimulate the nitrifiers, and we add deuterium also as an activity marker. After incubating, we prepare the samples to release the microcolonies, and then we use the Raman to identify nitrifiers and active cells, and then go on with MDA, PCR screening, and Illumina sequencing. So the reason that we have to do some sample preparation is here you can see that the native biofilm, and in here in blue and orange, you can see the nitrifier microcolonies. And so all of this other heterotrophs and so on. Now, this is not from the Hubble telescope, but this is actually the microcolonies after sonication. This is a composite, so they weren't all sitting here like this, but just to give you some examples. So you can see here, for example, the nitrite oxidizers in the center with the ammonia oxidizers around them. We have, in some cases, we find only the nitrite oxidizers, so they're not always together. And in some cases, we also have heterotrophs associated with these microcolonies. And the heterotrophs, this is not just a contamination, but actually heterotrophs are known to feed on nitrifier exudates. So they're really a part of the ecosystem. And also some bacteria are predators of nitrifiers. So within each of these separated microcolonies, we have essentially a mini ecosystem that we're really interested in exploring how this ecosystem is different from this ecosystem and this ecosystem, all within highly related organisms. And so we select who we want. We pick who we want using Raman-activated cell sorting. Right now, we're doing this manually. Taekwon is doing it, I should say. And what he does is he puts a sample into a capillary. You can see here under the Raman. And then he can measure each microcolony in the capillary. And then he looks either for the cytochrome signal. So nitrifiers have naturally a very high cytochrome signal. So he can select colonies based on the cytochromes or on the presence of the deuterium label showing that it was active in the enrichment, in the incubation. Once he's found the microcolony he likes, he can grab it with optical tweezers and then move it out of this area into the sterile end of the capillary. He cuts it off, and then he can spin it down into a tube, do MDA, do PCR screening, and it works. Yeah, so I promised you sequencing data, so here you go. So from the PCR screening of a single microcolony, here in blue, you can see the AMOA. This is the functional, the monooxygenase of the ammonia oxidizer. This is what we recovered from the PCR screening. When we did the sequencing, we were able to bin nitrosomonas, this ammonia oxidizer. We also see Nitrospira, which is a nitrite oxidizer over here. We also managed to sequence a little bit of Taekwon, but I won't go into those results today. But you can see that the PCR results and the sequencing results give basically the same AMOA. Now, if we recruit these reads to the closest sequenced genome, we get what we think is very good coverage. So 36% of the genome is covered from this nitrosomonas with 60 to 100% identity. And so now Taekwon has been very busy manually sorting based on deuterium content, based on cytochromes. And as part of a CSP project, we'll be sequencing 100 of these Nitrifier microcolonies. And we're really excited to see what kind of biological stories or outcomes we have based on this. And of course, this is great, but I told you that Raman is very fast, less than a second for a single measurement. This sorting process is very slow. Taekwon's very good at it, but it takes him about an hour to sort one cell out. So really, the major challenge that we are interested in tackling is to go microfluidics and make this whole thing high throughput. And this, we have a collaboration with Roman Stocker in the context of the ETOP program. And yeah, actually, there was a poster on Tuesday night by the postdoc of Roman, Kang-Soo Lee. So I preemptively will direct any really detailed questions about how this microfluidics works to him. But this looks very promising, and we're hopeful that this is going to speed up the whole process so that we can do this Raman-activated cell sorting really in a high throughput manner. So yeah, just to conclude briefly, basically I told you that in situ microbial activity can be studied with single-cell isotope probing in combination with FISH, and I hope that I've really conveyed that this is an extremely powerful method. It's really complementary to sequencing methods, and it's the best way to really directly prove a function. And from the nanosim story, I showed you that Bacteroides acidophacines and Akkermansia mucinophila are the major mucus degraders in mice, but that this activity is context-dependent, and that we were only able to see this based on this stable isotope probing approach. And I showed you that by labeling with deuterated water in combination with Raman, we're able to look at microcolonies and detect activity, and this sorting offers us direct genomic access to active nitrifier microcolonies and biofilms. So I'll just acknowledge that we have, this is actually only a subset of many good collaborations and also funding agencies that have enabled this research. And thank you for your attention.
So we have time for a few questions. Yes. Do we have a microphone?
So aside from phenylalanine, are there other molecules where you can actually tell where the label is going in some of these cells with Raman? Mm-hmm. Or can you only measure essentially fully labeled cells from non-fully labeled cells?
So phenylalanine gives the best signal, let's say. But it doesn't mean that you have to use a labeled phenylalanine to detect labeling. So unless you have some evidence that the cell is using a transporter to import phenylalanine, and therefore the phenylalanine, so the 13C content in the phenylalanine is not representative of the whole cell, that's the major assumption of this approach, right? So if for some reason the phenylalanine is coming from somewhere else than the other parts of metabolism, then using phenylalanine as a marker won't be good. But in most cases, what we've observed is that this is not the case. So when cells will produce new phenylalanine, and the 13C will go into it. There are other shifts in the spectrum, but none of them are really as strong as the phenylalanine. So we haven't explored that as a quantitative marker.
One more.
Yeah, so you mentioned that the Raman is extremely fast, but in the microfluidic platform, how many events can you analyze or sort per second?
I think that and I'm not the expert on the microfluidic side, but my understanding is that the Raman spectral detection, this is the slowest part of the whole thing. So The challenge we have more is how do we kind of slow down the microfluidics in order to get a good Raman spectrum and make a decision of where it should go. And we have different designs that we're playing around with a bit. But there are solutions to this.
Okay, if there's, oh, one more question.
What's your detection limit for the deuterium? Like, what atom percent do you need to actually be able to detect the cell being enriched?
Mm-hmm. We've done all of these measurements with the nanosims also, so that we know what the actual atom percent is in the cell, and we're down to 3 or 4 atom percent. So it's not as the Raman is actually extremely sensitive here. So, and it's more sensitive than it is with carbon. So, and the reason for this is that the CD peak is appearing in what's called the silent region of the Raman spectrum. And so we really have a very good detection limit there. Whereas with the phenylalanine, there's other materials at 960 that also give some Raman signal. So we can't have quite as good of a detection limit. But with deuterium, it's very good. So what I didn't mention is that if you use deuterium at high levels, this can be inhibitory. For eukaryotes, this is more of an issue. So for all of the microbes we've worked with, if you use deuterated water at 50% or lower, we don't really observe any inhibition. But for eukaryotes, this would be a problem. So we have to know what system you're working with.
Great. We'll give David another hand. Thank you.